like guanosine capping. Then splicing, where the non-coding introns are removed, and then you have the 3' tail. So all my work focuses on this last step, which is 3' tailing. So 3' tailing, or the pre-mRNA 3' end cleavage and polyadenylation, is a process which has two concerted steps, that is cleavage of the RNA followed by polyadenylation. This is essential for the stability of the RNA, its export to cytoplasm, and also to give it a translational efficiency. So what happens is the RNA is cleaved downstream to a hexamer sequence known as polyadenylation signal, and a long stretch of K-tail is added. So the long stretch of K-tail and this cleavage reaction, these all are orchestrated by a certain set of cis and transacting factors. Cis elements, or the sequence elements which helps in this recognition, are as such, and the major or the important ones are this poly A signal as well as the cleavage site. And these sequence elements are identified by a group of transacting factors which are categorized as CPSF complex, CF2M, CF1M, and CSTF complex, along with the poly A polymerase, which actually adds the A tail. So in this complex, we have the CPSF73, which is the endonuclease which cuts the RNA, and then you have the poly polymerase. So this is a canonical processing complex. Apart from this, we also have a non-canonical polypolymerase. This was identified in Dr. Anderson's lab. So here we have a non-canonical nuclear polypolymerase known as STARPAP, which identifies a set of cis elements, and it adds poly A tail to that set of RNAs. So the interesting thing about this polypolymerase is that it has been studied in various diseases like cancer, cardiac hypertrophy, and also in cellular processes like oxidative stress and apoptosis. So I will be more focusing on STARPAP, and also I will be talking some things about the cleavage site and poly A signal. So so there are several open questions at the 3' end of the RNA, and like how a poly A tail length of RNA is controlled, or how nuclease is activated. And through this thesis, I will be trying to understand how cleavage site is selected, or how cleavage site selection, or how it is happening, and how it is regulated, and also how RNA binding proteins are regulated, or are regulating this